through transcription factors?
Yeah, transcription factors. So actually, you guys, I forgot to record this, so I barely started recording right now. Whoops. Okay. Right, so in Archaea you have transcription factors, which are like proteins that are going to bind to the promoter, and then the RNA polymerase will come and attach to those transcription factors, and thereafter it'll be attached to the DNA. How does it happen in eukaryotes? How does RNA polymerase attach in eukaryotes?
Same as Archaea?
Yeah, same as Archaea. It's using the transcription factors. So in eukaryotes, you also have those protein transcription factors that attach to the promoter, then RNA polymerase attaches to the transcription factors. What's the difference between Archaea and eukaryotes and how that works?
Archaea have several transcription factors, Eukarya has many?
Yeah. So Archaea only have so many different transcription factors, and in eukaryotes you have, a truly ungodly amount, many transcription factors. And you might need several different transcription factors to all attach to the promoter at the same time before the RNA polymerase is able to attach. So it's just basically simpler in Archaea with fewer transcription factors needed. Okay. So you have your two strands of the double helix. One of them is going to serve as the template strand. What are you doing with the template strand in transcription?
That's like what you will be using to copy for RNA?
Yeah. So you're using that to build your RNA strand. So you're going to have your RNA nucleotides base pair with the template strand, and then RNA polymerase will add them to the new RNA strand that you're building. Then the other strand is the coding strand. Why do we call it the coding strand?
Because it's a sequence of the same RNA but are resulting of no, it's the sequence in the same as resulting RNA, but instead of the T, it replaces with a U.
Yeah, exactly. So they call it the coding strand because it has the same code as the RNA that you're building, or it has the same information, which is the same sequence of bases, except that in the coding strand you have T's, and in the RNA strand you have U's. Because your RNA strand is going to be base paired with your template strand, and the coding strand is just the other strand of DNA, so that would also be base paired with the template strand. If two things are going to base pair with the same strand, they have to have the same sequence of bases, or they wouldn't be able to do that. Except that, you have U's instead of T's in the RNA. Cool. And I guess we basically covered the RNA polymerase role, but I guess what is the process of how transcription goes? So I guess you start with your promoter, and you either have sigma factor or a transcription factor attached to that promoter. Then RNA polymerase will attach to that promoter based on the sigma factor or the transcription factor. Then what happens next?
It opens the double helix and adds
Yeah.
nucleotides to RNA strand.
Yeah. So it's going to separate the two strands of the double helix, so you don't need a separate enzyme to do that. The RNA polymerase can do it for itself. It's going to separate those two strands and start building an RNA strand based on the template strand. And it's just going to go until it gets to the end of the gene, basically. or the end of the operon. So then after you've done transcription, you have RNA processing. Which types of organisms do RNA processing?
Eukaryotes?
The eukaryotes, yeah. If we want to get technical, prokaryotes also have ways that they process their RNA, but it's not really as extensive, it's not as complicated, and it's not as important as it is for eukaryotes. So we mostly focus on RNA processing as a eukaryotic activity. So first you have the cap and the tail that would be added to your new piece of mRNA. What's the cap? What's the full name for the cap? Or I guess I should say, which end of the transcript do you put the cap on?
5 prime?
At the beginning.
Yeah, the 5' end. So we call it the 5' cap. Then at the other end, the 3' end, you put the tail. And what's the name for the tail?
Poly A tail?
The poly A tail, yeah. So on the 5' end, you have the 5' cap. On the 3' end, the poly A tail. What's the role of the cap and the tail?
The cap works backwards, modifying the G needed for translation.
So the cap is a modified, it's a modified G that's added backwards by a special process. But that you actually don't need to know. But you do need to know that the cap is needed for translation. The cap is used to attach to the ribosome in translation. Then what's the job of the tail?
It's protecting the mRNA from degradation.
Right, yeah. So eukaryotes are going to be constantly degrading any RNA that they find lying around in the cytosol. In order to prevent that from happening to their own mRNA, they add the tail to it, the poly A tail, just a lot of A's. And that prevents the enzymes in this cell from degrading that mRNA. So that's the first type of processing, adding the cap and the tail. Then you have splicing. So what is splicing?
Sequences that need to be cut out of the primary transcript.
Right, so that's where you have like some parts of that primary transcript that need to be cut out, and then the flanking parts need to be joined together. So in splicing, you cut out what needs to be cut out, and then what's left, you join the ends of it together, so you have a single strand still. Is an exon something that's going to be cut out, or something that's going to stay?
The exons you were keeping?
Yeah, it's for keeping. So it sounds like it's going to be cut out because it's an exon, which just sounds like exit, but it stays. So I just remember that it's the opposite of what I think it is. And then an intron would be what gets cut out. So in splicing, you're cutting out the introns and joining together the exons. Cool. Then translation. So we have, again, a number of molecules that contribute to translation. What's the job of the tRNA in translation?
It brings the right amino acids specified by each codon to the ribosomes.
Right. It's making sure that you have the correct amino acid brought into the ribosome. So it's going to pair with the codon, and then it'll have the correct amino acid attached to it for that codon. Then what's the role of the mRNA?
It divides into 3 base codons that specify the amino acid where it goes where.
Right. So the mRNA has the codons. Every codon is 3 base pairs, and it tells you which amino acid to put. So the mRNA is making sure that you get the sequence of amino acids right, and then the tRNA is making sure that for every codon, you put the right amino acid. Then what's the ribosome do?
Assembles on mRNA.
Right, it's going to attach to the mRNA, and then it's kind of the giant protein complex that's kind of coordinating this process and doing parts of it and then making sure that other parts happen right in a way. So the ribosome is what's going to be actually adding amino acids together to make the protein, and it starts at the the beginning of the mRNA, and it's going to make sure that it's always looking at the correct codon, and it has spots for the tRNAs to come in, so it makes sure that the tRNAs are able to pair correctly with the codons. So it's sort of the factory where all of this is happening. Then inside of the ribosome, you have rRNA. What does the rRNA do?
It catalyzes peptide bond formation between amino acids on tRNAs.
Yeah, exactly. So technically there's multiple pieces of rRNA, and there are multiple functions that those different pieces are doing. But the most important one is that the rRNA is actually catalyzing the peptide bond formation between amino acids. So if you had a ribosome but it didn't have any rRNA in it, it wouldn't actually be able to do translation. It couldn't build a polypeptide by adding amino acids together. It's the rRNA that's actually doing that part of building the polypeptide. And then our last protein here is the release factor. What does that do?
Release factor? It's a proteins bind separate polypeptides from tRNA and causes ribosome to dissociate.
Yeah, exactly. So the release factor kind of looks like a tRNA, but it is not. It's a protein. It's going to come in at the end of translation. It pairs, well, it doesn't technically pair with anything because it's not RNA, but it's going to come in when you get to the stop codon. And once it's in there, everything is going to let go of everything else. Everything will just kind of float away. And at that point, translation is over. So we generally would divide translation into the 3 phases: initiation, elongation, and termination. What basically happens in initiation?
Ribosome assembles on mRNA.
Yeah, exactly. The ribosome is going to put itself together on the mRNA, and then you have your first tRNA come in as well and pair with the start codon on the mRNA. So everything is basically just getting set up in initiation. How does the ribosome attach to the mRNA in a prokaryote?
By ribosome binding site.
Yeah, the ribosome binding site, which is also called the Schindel-Garneau sequence. So I just put both words on there because in 1306 we learn it as the Schindel-Garneau sequence, but then this book is using the ribosome binding site, or RBS, terminology instead. So I just used both. So that's just a specific sequence of in the mRNA, like at the very front of the mRNA, where the small subunit of the ribosome is going to attach in a prokaryote, and then the rest of the ribosome will assemble on that. In eukaryotes, it happens a bit different. How does the ribosome attach to the mRNA in eukaryotes? We actually already mentioned it with the RNA processing.
By the initiation factor?
You are going to have some initiation factor proteins that you need for this process to occur correctly, but those are actually not going to be on the test. It's something that's on the mRNA itself that the ribosome will attach to.
Oh, it uses the 5' cap?
Yep, the 5' cap, exactly.
Okay.
Cool. And then within the ribosome, you have your 3 sites where tRNA can bind. All right. And then those are going to be important for elongation. You're going to start out at the end of initiation, you have your first tRNA in the P site, which is the middle site. Why do they call it the P site? What does P stand for?
It stands for peptide?
Yeah, peptide. So that's where your peptide bonds are going to be forming in the peptide site, or the P site. So you have a tRNA starting out in the P site at the end of initiation. Then when you go to elongation, where you're actually kind of building your polypeptide, what's the first thing that's going to happen there?
A new tRNA enters base pairs with codon at A site?
Yeah. So you have a tRNA coming into the A site, and it's going to base pair with the codon there. So you make sure that it's the right tRNA coming in. What's the A site? What's the general job of the A site? Or I guess what happens at the A site? Sorry? Yeah, acceptor.
Yes.
Yeah, it's the acceptor site. So that's where new tRNAs are always coming in. Anytime a new tRNA comes in during elongation, it's always coming into the A site. So now you have a tRNA in the P site and in the A site. What's going to happen next?
It exits.
Sorry?
What happens after the tRNA?
Yeah, what happens after your tRNA comes into the A site and base pairs there?
The RNA
RNAs
receptor bond.
Yeah. You're going to have the rRNA catalyzing a peptide bond between the amino acid on the A site tRNA and the P site tRNA. Where does that, so that gives you basically the beginning of your polypeptide, because now you have 2 amino acids attached together with a peptide bond. So it's real little now, but it's technically now a polypeptide. Which tRNA is that going to be attached to? The P or the A tRNA?
The P.
The P. No, sorry. The A. Yeah, so after the peptide bond is formed, then the whole polypeptide is going to get transferred to the A site tRNA. And then what happens next?
Ribosome translocation.
The ribosome is going to translocate
down.
Yeah, exactly. So it's going to move along the mRNA. It needs to move by exactly 3 bases so that the codon on the P site switches to the E site, the codon on the A site switches to the P site, and a new codon comes into the A site. Then the tRNAs that were on the P and the A site are also going to get switched because they're still attached to those codons. So the P site tRNA now ends up in the E site. The A site tRNA now ends up in the P site after your ribosome moves. And then what happens next?
E site tRNA exits?
Yeah, the tRNA in the E site is going to exit. So the role of the E site is just to have a tRNA leave, or it's the exit site. So now you end up with just your tRNA in the P site, and since that used to be the A site tRNA, it has the polypeptide attached to it. So that's kind of one full cycle. And now you start again and do those steps over again. And that's basically going to continue throughout elongation. The ribosome is going to keep walking down the mRNA until it gets to what? What type of codon does it have to reach?
The stop codon?
The stop codon, yeah. And then when it gets to the stop codon, you enter the last phase of translation, which is termination, where everything is stopping. What happens when you get to that stop codon?
A release factor proteins binds, separates the polypeptide from the tRNA, and causes ribosome to dissociate.
Yeah, cool. So then your release factor will come in. There is no tRNA that will bind with a stop codon. So you're going to have a release factor protein come in instead. And once it's in there, everything lets go. And then the process is done. Okay. So let's see, I guess we didn't mention the anticodon. What's an anticodon?
Three bases that pair with the mRNA codon.
Yeah. And where would you find an anticodon? What type of molecule has it?
The tRNA.
Yeah, exactly. So every tRNA has an anticodon that's going to base pair with the codon. So on one end of your tRNA, you have the anticodon. On the other end, you have the amino acid that it brings in. Cool. And I think, do we mention a start codon? What's a start codon?
A start codon is which signals the ribosome to start translation.
Right. So in initiation, you're going to have the ribosome assemble so that the start codon is in the P site, and that's where everything's going to start. So you're going to have your first tRNA, the initiator tRNA, come in and pair with the start codon, and then you'll go from there. So it's really important that the ribosome always gets the start codon into the right location, because otherwise you might end up reading codons the wrong way. Cool. So that's the basic process of translation. But there's also one more thing that kind of happens in translation that ends up being important for organisms and for their mutations, which is co-degeneracy. What is co-degeneracy?
Multiple codons can specify the amino acid.
Right. So where you can have multiple codons that all mean the same amino acid. So for every amino acid, you're only going to have one codon. That means, for for every codon, you're only going to have one amino acid that it means. But for every amino acid, you could have multiple codons that all mean that same amino acid. Yeah. And that's because when the tRNA is attaching to the codon, it actually might not attach perfectly right, because the anticodon on that codon is kind of curved. Sorry, the anticodon on the tRNA is kind of curved. So the last base in the anticodon is kind of pointing off to the side, and it's not going to base pair right with the last base on the codon anyways. So it can kind of pair with multiple codons. So all of those codons are going to end up meaning that amino acid for that's carried by that tRNA. Right, but actually you don't need to know the basis of co-degeneracy. You would just need to know that it means that you have, for every amino acid, multiple codons that all specify that same amino acid. And then you would also need to know why that's important for organisms.
It reduces the impact of mutations if they occur at the last base codon.
Right. So that means that if you have a mutation like at that last base in the codon, it's probably okay. There's not going to be an effect of it probably, because you can still have the same tRNA bind there. It might still be a codon that codes for the same amino acid. So if you have a mutation at the first or the second base in a codon, that's probably bad. I mean, it probably at least means that the amino acid is going to change, so it's going to have an effect. But if you have a mutation in that last base of the codon, it's probably not going to have an effect, or it's much less likely to have an effect. You might still end up putting the same amino acid into your protein, so that there's no functional effect of that mutation on the proteins that you're making. They still look exactly the same. So it's kind of insulating organisms from their mutations. Okay, so once you've done translation, technically you don't have a protein yet. If we want to get real technical, it's still technically considered a polypeptide because it's not yet necessarily functional until it folds. Once the polypeptide is fully folded and functional, then we call it a protein. What's the molecule that would help a protein fold correctly?
The chaperone?
Yeah, the chaperone. So how does the chaperone help a protein fold?
It ensures correct folding. Mostly they prevent wrong folding, space things up, and can also recall denatured proteins.
Yeah, exactly. So your chaperones mostly are just going to be making sure that the protein folds really quickly and that it doesn't end up folding the wrong way. So it doesn't necessarily make it fold the right way, but if it notices that it's folding the wrong way, it's going to stop that from happening. And then it ends up folding the right way once all the wrong ways are kind of blocked. If you do have cells that are exposed to like too much heat or too much pH, something like that, sometimes you can have chaperones that actually refold the proteins that were denatured. So once your protein is folded, now that's a functional protein, that's a protein. But you still might need it to get sent to the right spot. If you're a prokaryote, where would you be sending your proteins if you're going to send them to some specific location?
For prokaryotes, they must be
outside the cell.
Yeah, true.
Yeah. Or I guess technically, if you're gram-negative, maybe the periplasm. But for prokaryotes, there's nothing really special going on the inside for the most part. So if your protein needs to go somewhere special, it's going to have to go outside of the cell, either to the periplasm or actually outside of the cell. For any protein that needs to go outside the cell, what does it need to have to make sure it gets there?
a signal sequence?
Yeah, the signal sequence, which is what?
It targets proteins to cross membrane in prokaryotes. Lets proteins interact with secretion system proteins.
Yeah, it's going to allow those proteins to interact with the secretion proteins that can put them outside. So it's going to send them out to the outside of the cell. And what does it actually look like? Like, what's it made out of? What's the whole protein made out of? Or what's the building block for the protein?
Is it amino acids?
Yeah, amino acids. Yeah.
Oh.
So it's a specific sequence of amino acids that's like at the front of the protein. And every protein that has like amino acids at the front in that specific order, that's the signal sequence. So those proteins are all going to go outside the cell. If this is a eukaryote, then you kind of have a lot of places where you might need to send your proteins because you have a lot of different organelles inside the cell. And how are you going to make sure that those proteins get to the correct location?
They use the proton motive force.
Let's see, that's going to be for, I'm not sure if there are any eukaryotes that use the proton motive force, actually. There might be some microbial eukaryotes that do, but I'm not specific. I don't know. But the proton motive force is going to be mostly relevant in prokaryotes, and they're definitely going to use that as like a source of energy to send things out of the cell. For eukaryotes, they might use ATP. They might use some type of ion gradient. So I guess that might be a proton motive force. I guess you might call it that, as a source of energy. But how are they going to actually target things to go outside of the cell, or to go to a specific organelle in the cell?
I don't know that one.
For sure. So for eukaryotes, you're going to use some type of tag. It could be a sequence of amino acids, like similar to the signal sequence, but different because it's for eukaryotes. So for instance, if you need to send a protein to the nucleus, there's a specific sequence of amino acids that will send that protein to the nucleus if it has it. But you could also have like tags that are added to the protein after it's been made. Those tags would usually be made out of sugars. So in the Golgi apparatus, you'll be attaching different tags to proteins, different sugars, different modified sugars, and depending on what like combination of sugars that protein got, that's determining where it's going to go. So for eukaryotes, you might have a sugar tag, or you might have an amino acid sequence that are going to be sending that protein to its destination. And for prokaryotes, you just have the signal sequence, the sequence of amino acids that will send it to the outside of the cell. So that's chapter 4. Cool. So it looks like we're going to have time for chapter 5 probably. I'm guessing, I don't know, because you guys probably haven't had much of a chance to get into chapter 13. So maybe I'll wait for that one, and we can probably go over that in our next review session. And now we can go over chapter 5. So, what does growth mean in microbiology?
Growth is the increase in the number of cells.
Right. That just means that the cells are reproducing, they're dividing, and the numbers of cells in that population is going up. So for most creatures, growth means that the actual organism is getting bigger. For microbes, it just means that you're ending up with more of those organisms, and the population is getting bigger. What's the generation time?
How long it takes for the cell to divide?
Yeah. So for any given microbe, the generation time, how long it takes for that cell to divide, which is going to be different for different microbes. So the population of microbes in general is going to double every time that you have, like, every time that one generation time is up. So if the generation time is 4 hours, every 4 hours that population is going to double on average, because every cell will be dividing every 4 hours. What does death mean for microbes?
Just the population starts decreasing.
Not necessarily that it starts decreasing, but that you don't have more growth. And this because a lot of the time it's kind of hard to tell the difference between a microbe that's alive and a microbe that's dead. Sometimes a dead microbe will actually kind of have its cell like break open, it'll lyse and it'll be destroyed, and so then you would be able to see that the population decreases. But most of the time they're just going to be sitting there and they're dead now, but you can't tell. So they still look like they're contributing to the population even though they're dead, or you would still probably count them. So we're defining death for microbes as just you tried to grow it and it did not grow. No more growth. And yeah, just because it's hard to tell when they're dead, when they're actually dead. Like for any other organism, you can pretty much look at it and see if it's dead or alive. But for microbes, since they're so small and so simple looking, it's more complicated to tell if they're actually dead or not. But you can definitely tell that they're dead when you notice that they didn't divide. Okay. Then what's a colony forming unit or a CFU?
It's used for cell clumps that can form colonies.
Yeah, it's clumps, either single cells or little clumps of cells that will form a colony. And the reason why we have that term is because a simple way of counting bacteria is just to get your bacteria, like kind of mix them up in the sample that you have, and then spread them out on a plate, and then wait to see who grows into a colony. So ideally, what you want is for the bacteria to all be separated from each other, and then you spread them out kind of thinly, and then every single bacterial cell turns into, a little colony of cells after some time has passed and it's grown. But you don't actually know if that colony started with one cell or with a little clump of cells, because it can be kind of hard to detach bacteria from each other. Some of them are really sticky. So it might have been that one cell made that colony, or it might have been a little clump. So we say colony forming unit. And then you just count up the number of colonies on your plate, and that's the number of colony forming units that you had. Ideally, each one is a single cell, but that might be a clump. Cool. Then we have the decimal reduction time. What's that?
the time it takes for 90% of microbes to die in presence of an agent?
Yeah. So if you are exposing those microbes to something that's going to kill them, the decimal reduction time is how long you have to expose them in order for 90% of them to die, which would mean that when you try to grow them later, you only get 10% of the growth that you were expecting to have, given the population size. So that means that if you're using some type of agent to kill microbes, you got to make sure that they're exposed for enough time. Usually you want to do multiple decimal reduction times actually, because the first time you do it, there's still going to be 10% left. And since the populations would probably be like millions in size, that's still a lot of microbes left over. Cool. So if you're doing batch culture, you're growing your microbes in some type of container, like a flask or something, you're going to have the 4 different growth phases that you see. What's the first growth phase?
The lag phase?
The lag phase, yeah. What's happening there?
The population initially doesn't grow.
Yeah. You don't see any increase in the population. And why is that happening?
Cells aren't making stuff for division?
Yeah. So the idea is that you just started this culture, and the cells aren't used to growing on this media, but they can do it, but they're just not used to it. So there's some things that they need that they don't have yet, and they're going to need to make those things. So the lag phase, they are making all those things that they need to be able to grow on this media, in preparation for cell division. Then once they've got everything they need synthesized, now you would move from the lag phase into what?
To the exponential phase?
Into the exponential phase, yeah. What happens in the exponential phase?
The population doubles.
The population doubles according to the generation time. So every generation time, you'll have the population having doubled. Why is that happening?
Well, that's when the cells are most active and healthy.
Yeah. The cells are very healthy, they're very active. And the reason why they're so healthy and active is because there's a lot of nutrients available in the media, and there's not a lot of waste products. So you started with a small population and a bunch of nutrients and no waste products at all. And as the population grows, it uses nutrients and it makes waste products, but you still don't have very much waste products and you still have a lot of nutrients left. So the population keeps growing, doubling every generation time, and the cells are in real good shape. And as your nutrients get consumed and your waste products build up, what's the next phase that you're going to enter?
The stationary phase?
The stationary phase, yeah. What's happening there?
The population remains constant. They're not.
Yeah, you don't have much of a change in the population size. So you don't have as many nutrients left, so the cells are not able to divide quite as quickly. Also, you have a good bit of waste products built up now, and those are usually toxic, so the environment's not quite so nice as it used to be. So the cells are kind of starting to die, some of them, and as new cells are born by cell division, old cells are dying. And those two processes kind of equal each other out, so overall, you see that the population stays constant. Of course, if you're looking at total cell numbers, that's going to continue going up because all those dead cells are still there. But if you tried to then kind of grow those cells, you would see that the amount that grow is the same as you move through the stationary phase. You're always having the same amount of cells that are actually able to grow. Then what's the next phase?
That would be the death phase.
The death phase, yeah. And what happens there?
Well, the population will decrease.
Yeah. So now the population is going down, or I should say the population of viable cells that are still alive is going down. It's actually going to go down exponentially as nutrients run out and waste products are starting to get to a real high concentration. It's becoming kind of a hostile environment now. There's not a lot of food and there's just a bunch of crap. So the microbes are going to be dying exponentially. Which means that you might look and see that the total number of cells is still the same, but if you tried to move those cells onto new medium and grow them, you would see that most of them are not able to grow, so they're dead. So out of all those 4 phases, what's the one that you would like to do an experiment during? Probably.
The
exponential phase, right? So that's when your cells are nice and healthy and very active. So you usually don't want to do an experiment with cells that are sickly and, you know, kind of marinating in their own waste and not getting enough nutrients. Then we've got our different types of media. So what's defined media?
When you know exactly what is in it and how much.
Right. You know exactly what's in there and what concentration for everything that's in there, which probably means it's synthetic. Everything that's in there, you or someone else added in there at a known concentration. And then that's kind of the opposite of a complex media, which is what?
You don't know exactly what's in it or how much.
Right. So that's where you don't know exactly what's in there, or maybe you don't know how much is in there of each thing that's in there, which would mean you did not just sit there and make it, adding every individual component. You got that part of that media, at least, is coming from a natural source. So you don't know exactly what all is in that natural source. So every media is going to be defined or complex, and then it can have also other types as well. What's an enriched media?
Where extra growth factors are added.
Yeah. So that's a specific type of complex media where you added some natural substance that's really full of growth factors. And the most common one is going to be FBS, or fetal bovine serum, which is just cow's blood, well, you know, fetal cow's blood, without the clotting factors, so it doesn't clot. If you have bacteria that really need a lot of growth factors, then you want to grow them on enriched media with the FBS so that they can get all the growth factors they need. You don't know exactly what growth factors are there or how much is there. It's just an easy way to add a lot all at once. What's a selective media?
It contains or leaves out stuff that inhibits growth of some microbes or favors growth of others. Not all microbes are expected to grow well.
Right. So the selective media is what you're going to use when you're trying to grow one type of microbe specifically, or if there's a type of microbe that you're specifically not trying to grow. So if there's someone that you're trying to go specifically, then you might put in a nutrient that you know that that guy can use that a lot of microbes cannot use. So that microbe is going to be favored to grow in that media with the that only it can use. Or if there's a type of microbe that you want to keep out, you might put in a toxin that's only toxic to that type of microbe. Or maybe you want to grow like acidophilic microbes, so then you would put a low pH media. Anybody who's alkalophilic or neutrophilic, the pH isn't going to be right for them, they're not going to grow. So in the selective media, you have something that is limiting who all can grow on it. either favoring some microbes or inhibiting others. Then what's an enrichment media?
Selective media that should increase growth of rare or slow growing microbes that will otherwise would be crowded out.
Right. So that's a special type of selective media where you have some microbe that just is really hard to get growing, either because there's like one of those like one cell in, like, you know, if you have a sample of 1 million different bacterial cells and only one belongs to the species that you're trying to grow, it's going to be hard to get that guy to grow enough to where you can actually find his colony. Or if he grows really slow, like Mycobacterium takes like months to grow, that's going to be difficult for a microbe like that to grow to a size where you can actually isolate a colony in the midst of all these other microbes that are more numerous or growing more quickly. So with the enrichment media, you're specifically super targeting that one microbe that you're trying to get to grow. You're probably making the media pretty hostile to other microbes and pretty favorable just for that microbe. And then finally, we have the differential media. What's that?
It contains indicators that lets you know something about the microbes and their metabolism.
Right. So that is a media that is telling you something about who's growing on it. So maybe it's going to change color. Well, usually it will change color. Maybe it changes color when the pH goes down, and then you know that a microbe that produces an acid, or maybe the color changes when a specific like iron deposit forms, and then you know this bacteria is metabolizing iron. There's just something about that media is going to change when the microbe is growing on it, and that produces usually a color change, something where you can just look at it at a glance and know this type of metabolic process is happening in these bacteria growing here. So that's pretty useful because it lets you kind of grow the microbes and test them at the same time without taking extra steps. Okay, and we had like four different variables that affect microbial growth that are not just how many nutrients are around. What are those variables?
Temperature,
The temperature,
pH, oxygen,
pH.
and water.
Yeah, how much oxygen is there and the water presence. Right, for water, that's actually kind of like two, both how much water is there and how many solutes are dissolved in that water. Then we have a bunch of different microbial groups, and a bunch of them are going to be on the test, but not all of them, happily. So what's a psychophile?
To be in cold temperatures, can tolerate warm temperatures.
Right, that's a microbe that likes it cold. Then a mesophile.
They're like medium temperatures, most relevant microbes for us in terms of our health.
Yeah, so they're gonna like a medium temperature, kind of a temperature that's like the room temperature or like your own body temperature. So since those are the guys that like the temperature that's inside of your body, those are the ones that are able to grow inside your body. They're either good or bad, or maybe neutral. So your microbiome is going to include mesophiles, also pathogens that would make you sick. They're usually going to be mesophiles. They have to be able to tolerate it at least inside your body. What's a thermophile?
Like
Likes to
heat.
be
Right, they like it hot. The thermophiles like it hot, and then hyperthermophiles like it really hot. Technically not on the test. Then what's an acidophile?
It likes low pH or acidic environment?
Yeah, it likes it acidic. And if it's acidic, that means the pH is low. So low pH acidic, high pH alkaline or basic. And what's a neutrophile?
Likes medium pH,
It's gonna
neutral
like
environment.
Yeah, it likes a medium pH, which would be a neutral pH, which would be like your pH. Then an alkalophile.
Likes alkaline environments with high pH or basic environments.
Yeah, so that's gonna like a high pH, which would be an environment that we call alkaline, or we could also call it basic. Then a halophile.
Needs a lot of salt.
Right. It likes to live in a place where there's water, but that water is full of salt. Like the ocean. So in the ocean you would have halophiles. Then what's an osmophile?
Needs high solute.
Yeah. So the technical definition is that that's an organism that needs to have high solutes in its water. So it needs to live with plenty of water, but the water needs to have a lot of solutes in it. Salt is a type of solute. So technically a halophile is also a type of osmophile. But since we already have like the word halophile and there's usually not that many different solutes in waters that interest us, we usually use the word osmophile to mean some microbe that likes a lot of sugar dissolved in its water. So technically a lot of solutes, but especially a microbe that likes a lot of sugars. So a microbe that's able to actually grow in like honey or jam, that's going to be an osmophile. Most microbes cannot grow there because there's just too many solutes, which is why you can leave your honey unrefrigerated. Then what's a xerophile?
Microbes that need dry environment.
Right. A microbe that likes no dry place, like the desert. And then an aerobe.
Microbes that do aerobic respiration.
Right. Any microbe that can do aerobic respiration is counting as an aerobe or an aerobe. And then we have 3 different types of aerobes. What's an obligate aerobe?
Needs to have oxygen O2 to grow.
Yeah, that's a microbe that has to have oxygen. It has to do aerobic respiration for energy, and it cannot substitute a different process. So if it doesn't have air, or I should say oxygen, then it is not going to grow. Then what's a microaerophile?
Can tolerate atmospheric oxygen levels.
Right. So that's an aerobe doing aerobic respiration. But if it's actually just out in the air with like 20% oxygen in the air, that's too much oxygen for it. It's going to die. So it doesn't have the right enzymes, or I should say it doesn't have enough enzymes to detoxify a lot of reactive oxygen species. It can only detoxify a little bit at a time, which means that it can only be exposed to a little bit of oxygen at a time. So you would find that kind of at the boundary, maybe in the water, at the boundary between like the part of the water where there's a lot of oxygen and the part where there's not. Just kind of where there's a little bit. Then what's a facultative aerobe?
they can do a aerobic respiration. Instead, they use fermentation.
So that's going to be a microbe that can do aerobic respiration but doesn't have to. So that means maybe it can do fermentation, maybe it can do anaerobic respiration, it can do something else instead. So if there's oxygen around, it'll do aerobic respiration and make the most energy possible. But if it ends up in a place where there's not oxygen, that's okay. It'll switch to fermentation or anaerobic respiration, and it has a backup plan. Cool. So those are the aerobes. We also have the anaerobes. What is an anaerobe?
Oh, that's the one that can do aerobic respiration, right?
Right. Yeah. No aerobic respiration. So that means they do fermentation or they do anaerobic respiration instead. What's an obligate anaerobe?
They can't tolerate oxygen, which
Right.
they won't grow or they'll die.
Yeah. Yeah. So an obligate anaerobe must stay in a place without oxygen at all times. It cannot handle oxygen. That means that it does not have any enzymes that would allow it to detoxify reactive oxygen species. So if oxygen comes into its environment, reactive oxygen species will form and it'll die. Then what's an aerotolerant anaerobe?
They can't tolerate oxygen, which they'll have enzymes they need to detoxify species oxygens.
Yeah, exactly. So they're not using the oxygen for aerobic respiration. They're getting their energy from the fermentation or anaerobic respiration. But if there is oxygen around, that's okay. They have enzymes to detoxify the reactive oxygen species, and they'll be fine. Okay, so out of all those groups, what are the two that are most common to contain human pathogens?
Non-halophiles and halotolerant. Halotolerant.
Yes, you would find human pathogens in the non-halophiles and the halotolerants. We don't have a lot of salt, but actually our skin does. On our skin, we'll at least be able to tolerate salt. But if they're also going to get you sick, they need to tolerate like low salt and salt. So you don't have any halophiles that really get you sick. It's going to be non-halophiles or halotolerants. Who else? What other group would include human pathogens?
Neurophiles.
Neutrophiles, exactly. Your pH is 7.4, which is a very neutral pH. So if an organism is going to be living inside of you and making you sick, it's going to need to be happy at that neutral pH. It's going to need to be a neutrophile. And one more group that contains human pathogens.
Is it alkalophiles?
No, it's going to be not an alkalophile. That would, I mean, technically 7.4 is a little bit alkaline. It's a little bit above neutral, true neutral, but it's not like far enough for an alkalophile to really be happy, most of them. And there's another group. It's not a pH group.
Is it a mesophile?
Yeah, mesophiles. Mesophiles like the medium temperatures, which includes room temperature and your own body temperature. Yeah, so mesophiles will be happy to grow inside of you. A thermophile, on the other hand, is going to find you too cold. A microphile is going to find you too hot. So only a mesophile will be able to make you sick. Okay. Why is it that we have some microbes that will die when it comes around? What makes oxygen so dangerous?
Because it can be in the wrong format.
Because it can what?
It can be in format.
Oh yeah, it can be in the wrong form. So O2, that's fine, not toxic at all. But reactive oxygen species, not so fine. Like hydrogen peroxide, that's a reactive oxygen species. So anytime that you have oxygen around in the presence of organisms through their metabolism are always going to create some amount of reactive oxygen species. And that is just going to be very reactive, like the name says. It's reactive. If it reacts with your proteins, they may lose their function. If it reacts with your DNA, it may be damaged. So if you have a microbe that's exposed to these reactive oxygen species and it doesn't have a way to detoxify them, it will inevitably die once its DNA is too damaged, once its proteins are too damaged. So if you have the enzymes to detoxify those reactive oxygen species, basically just reacting them until there's something that's not toxic anymore, then you can be safe in the presence of oxygen. But if you don't have those enzymes and oxygen is converted to ROS, then you're gonna die. Okay, what's a disinfectant?
It kills most of things, not all except endospores, and is used in surface.
Yeah. Disinfectant, first, is killing most stuff, but not all the stuff. And second, you're putting it on a surface or an object. So especially things that are resistant, that are like kind of hard to kill, they're gonna probably make it. Like an endospore, probably gonna make it. Some viruses, probably gonna make it. Coronavirus, probably not gonna make it actually. It's not one of those hardy viruses, it's a fragile virus. Lucky us. Then what's an antiseptic?
It's non-toxic and used for tissue.
Right. That is something that will kill microbes, but will not hurt you, which means you can put it on yourself. So alcohol would be a good example of that. So some things are going to fall into both categories, like alcohol is an antiseptic and a disinfectant. But then you have other things like Lysol or bleach. Those are disinfectants. They are not antiseptics. Cool. Then why would we autoclave something rather than boil it or stick it in the oven?
It's the most common way to sterilize through heat.
Right, it's the best way. Of all the ways that we can sterilize something using heat, is the best way, which is why it's like the most common way to do it. So what makes autoclaving something better than roasting in the oven?
It creates heat under pressure.
Right, you have heat. And one more thing about it.
It activates a higher temperature than just the boiling temperature of water.
Right. So when you have like pressure and you're creating heat and you're creating pressure, you can get water over the boiling point. Normally, if you start exposing water to heat, it'll go up to the boiling point, then it starts boiling, then all your water is turning into steam. It won't go hotter than the boiling point. If you have water that's hotter than the boiling point, it's steam, it's not water. The only way to prevent that from happening and get the water's temperature up above the boiling point is to put pressure on it so that the steam is not able to form. So in an autoclave, you have a lot of pressure, then you start heating your water, it actually goes above the boiling point because it doesn't turn into steam. So that means you can get it real hot, and that makes it better than boiling. Technically, you can get an oven real hot too, though. So there's something else about the autoclave that makes it better than an oven.
It doesn't denature prions.
That is true. That is true about all those things actually, except for burning. So autoclaving is real good. It's basically going to sterilize things, but if there's prions, they will still be there and they will still be infectious. Luckily, those are not very common, but if somebody is like infected with a prion disease, you can't just autoclave like the stuff that you used to treat that person because the prions will still be there. But it's actually because autoclaves use moist heat. In an oven, you just have like the air, you're heating the air, and then that heats the thing inside the oven. In an autoclave, you have water that you're heating. So it's a very wet environment. And water is just going to transmit heat better than air does. Air is actually an insulator, which is how like the double paned windows work. They have like a pane of glass, then a little bit of air, then another pane of glass, and they prevent like temperature changes because of that air is insulating. So if you put something in an oven, the heat that the oven is going to be transmitted to that thing super well. It'll transmit, but just not super well. If you have that thing in an autoclave, the heat is going to transmit to that object really well because it's in a very wet environment. So it'll actually end up getting hotter when you autoclave it than it would in an oven at the comparable temperature. So autoclaving is better than an oven because it's moist. Autoclaving is better than boiling something because it has that pressure and you can get the temperature higher than the boiling point of water. But if you have prions around, you actually need to buy like a special enzyme autoclaving for you. Okay, and then finally we had the suffix cidal versus the suffix static. So what's a microcidal agent?
It kills microbes?
Kills the microbes, yeah. And then what's a microstatic agent?
Prevents microbes from growing but doesn't kill them?
Yeah, it's going to stop them from growing, but technically they are not dead. So how would you be able to tell that the microbes aren't dead when you used a microstatic agent? How would you kind of check to see that the microbes are dead?
They use cryoprotectant?
Let's see, so that's going to be if you're like freezing a microbe, like a sample of microbes, and you want to make sure that they don't die, you're going to put cryoprotectant in there with them. So they don't freeze, they just get real cold, and then you can store them. So that technically would be a microstatic condition. They're not dead with the cryoprotectant, even though they're frozen. And then you know that they didn't die is when you take your sample back out of the freezer and you take it out of the cryoprotectant and you put it on a plate, you see that it does what? If it's not dead, what's it going to do on the plate?
Oh, the microbes will still grow?
Yeah, exactly. They're going to grow. So if you had exposed your microbes to a microcidal agent and they died, then you can like scoop up a sample of those dead bodies and put them on a plate and they will not grow. If you expose your microbes to a microstatic agent, you might see them, they're going to stop growing, so the population isn't going to go up, and you might think, well, they're dead, they're dying. But if you try to grow them, then if you scoop up a sample and put them on a plate, they will grow. They will start growing again. So static means they stopped growing, but they are not dead. They will grow again if you try to culture them. Microcidal means they are dead. They will not grow no matter what you do. Whew, okay, cool. So that's chapter 5. Cool, we got through it. Is there anything else that you guys wanted to ask, or you guys wanted to ask?
What's the basic process?
The basic process of what?
For
for transcription?
I think so.
Okay.
Yes, transcription.
That's cool. Right, so the basic process there is that you're going to have your promoter. If this is bacteria, sigma factor is going to attach to the promoter. If this is archaea or a eukaryote, a transcription factor, one or more transcription factors will attach to the promoter. Then RNA polymerase will be able to attach to the promoter, either using the sigma factor or using the transcription factors. So that's a start. Then RNA polymerase is going to separate the two strands. Then it's going to start building an RNA strand using the template strand of the DNA. So basically, you're going to have nucleotides coming in, RNA nucleotides, the RNA polymerase. They're going to base pair with the template strand, and if the base pairing is correct, then RNA polymerase is going to add them to the strand that it's building. And it's just going to go moving along the DNA, opening it as it goes, adding new nucleotides to the strand it's building. Then eventually it's going to get to some type of termination sequence that tells it to let go, and it'll let go. And then transcription's done.
Okay. Thank you.
For sure. Does anybody have any other questions? All right, sure. And we're about out of time as well, so that's perfect timing. Cool. So thank you guys for coming, and we can go over chapter 13 next week, next Monday. And yeah, I hope it was helpful, and I wish you guys happy studying and stay safe.
Thank you.
Thank you.
For sure. All right. Good night, you guys.
Bye.
Bye. Good night too.